Okay, so first of all, I would like to thank the organizers for giving me this opportunity to be here. Especially, I have literally 50 meters to walk from my office here, so there's no excuse. So today I'm going to tell you about something different from what we normally do, which is on this work on the LINE-1. And the work is spearheaded by a really talented postdoc in the lab, George, who's now just started his lab at Princeton, and he's also helped by Hong Mao, who is also another postdoc in the lab. So as this audience would know, that in the early 2001, the first human genome was sequenced. And one of the very interesting observations that came of it is that protein coding genes only account for about 1% of the genome. This is quite remarkable. You think about it, most studies in the last few decades have been focusing on genes. And 48% of the human genome consists of single copy sequences. On the other hand, the remaining half of the genome are made of what we call repetitive elements. And there are several major types of these elements. And these include long interspersed nuclear element, the lines, short interspersed nuclear element signs, human endogenous viruses, HERVs, DNA transposon, as well as tandem repeats. So far our lab has been studying telomeres that belong to these tandem repeats. And today I'm going to tell you about some of the latest work on one of the classes of the lines. And that is the LINE-1. So why LINE-1? So essentially, LINE-1 is a very short motif. It's about 6 kilobases in length. So it encodes 2 open reading frames. Open reading frame 1, which encodes an RNA binding protein. And open reading frame 2, which is the actual retrotransposition, is responsible for writing the LINE-1 into the genome. And this also has an endonuclease domain, which becomes important later in the talk. So why is this important? As you can see in the last diagram, the genome has about 17% of the LINE-1 accounts for 17% of the genome. That's quite remarkable. It's almost a fifth of our genome. So, and what's also special about this LINE-1 is the only known autonomously active transposable element in humans. So it can still jump around as we speak. So this activity, you can imagine, if it's not controlled properly, it can lead to severe gene disruption and rearrangement, and has been known to cause human diseases. So a couple of years ago, we started thinking about this, and the first question we started asking is how does LINE-1 spread throughout the genome? So the molecular mechanism of this process is not known, but I'm going to kind of put together kind of what is currently known based on a lot of functional data. So in this life cycle, LINE-1 starts with its transcription by RNA PO2, and this gives what we call the LINE-1 mRNA. This mRNA then gets exported to the cytoplasm, where it gets translated by the ribosome to give functional ORF1 and ORF2 proteins. So these proteins like to bind their own mRNA to form what we call the LINE-1 RMP. And this RMP then gets re-imported back to the nucleus, and for simplicity, I'm only showing the reverse transcriptase subunit here. And in the nucleus, it recognizes a very simple sequence motif, just 6 nucleotides, TTAAA. And what it will do, it will use this endonuclease domain to cut the opposite strand. You can see these are the multiple T's that is exposed here. And it binds to this own mRNA that poly-A sequences here. This can then really specifically base pairing with the poly-A tail of the LINE-1 RNA, and therefore allowing it to start reverse transcribing this own cDNA. This is a process we call target brand reverse transcription, or TBRT, and thereby the first strand is synthesized. The first question we wanted to ask is how this target DNA is recognized and restructured by the reverse transcriptase during this process. So here it only synthesized 1 strand. In order to complete the insertion, the opposite strand needs to be nicked, which is we call the top strand, and the second strand needs to be synthesized. And also, currently, what was not known is whether ORF2 complete this insertion by nicking the top strand and synthesize the second strand. And thereby, you get a complete LINE-1 insertion. So I'm going to hope to convince you some of our latest work today allows to give some insight into these questions that I pose here. So as biochemists and structural biologists, the first thing we try to do is to reconstitute this process in vitro. So to do this, we purify the ORF2 protein from insect cell, you can see here, and then we devise what we call the TBRT assay. So in this assay, we give the enzyme a target DNA, and this is actually derived from a known insertion in a patient, where this insertion happened in a factor A gene that called hemophilia disease in this patient that was described in the late 80s. So when we then give this enzyme DNTPs, if it's active, you can imagine it will cut this bottom strand and start synthesizing this first strand. So when we did that, what we use is substrate DNA, where we have a label on the bottom strand. We can monitor this process, and the projector is working. But what probably you wondering, there's no product here. It's just because the enzyme is not working. So this is showing us the enzyme itself is not very efficient at doing this step. So the next step of this reaction is obviously this bottom strand nicking. So we thought it maybe is the limiting factor. So we asked whether we can bypass this first strand cleavage by giving the enzyme a new substrate, where instead of giving this substrate, we give it a new substrate where this is pre-cut, whether it can do this reaction. Indeed, when we did that, we have robust activity. We'll see this product being synthesized on the top part of the gel. So with that, we then asked whether this sequence is actually specific for its activity. So what we did is we replaced the sequence with a GC-rich sequence, which is shown here. And you can see here again, we have no activity. So this suggests that this consensus motif is very important for its activity. So with all of these two boxes in our hands, we can then reconstitute this process biochemically and structurally. So to do this, again, we give the enzyme a poly-A RNA that can prime TBRT, and then we give a target DNA with a cut bottom strand, and then we give a DTTP. The enzyme will start the reaction and store due to this is chain terminator nucleotide. So with this, we solve the structure by cryo-EM to below the 2.5 angstrom resolution. This is 2.3 angstrom. And I'm going to walk you through the structure as we go. So this is an overview of the structure when the enzyme is in gray. I'm going to kind of highlight the domains in the subsequent slides. And the DNA is shown in blue and the RNA is in red. And if it turns around, you can see the DDTTP is in the active site. You can see here. So when George first showed me the structure, I thought, okay, we need some sort of analogy you can explain the structure. So I thought it looked like a handbag or a basket. And this is like a handle of the handbag, and this is the body of the handbag that encloses the active site and the DNA RNA duplex. So the ORF2 protein itself consists of 5 domains. So it has, and this is just a cartoon to go with the structure. So it had an endonuclease domain that I already highlight. Interestingly, in our consensus structure, this is very flexible. So you can see here, this is 2-dimensional class averaging of our data. So the core that you see on the left is right here, and this endonuclease domain is kind of moving around. So we also have a reconstruction where we resolve it at low resolution, but you just keep in mind that this is really, there's many conformation of this domain. And this is followed by the N-terminal extension that form this handle of the basket. And then the catalytic retrotransposition domain that has the active site that's incorporating this DDTTP. And then we also have a polymerase subthumb domain, and this is also a special for RT. This is very specific for polymerases. And LINE-1 also have a domain that's called the wrist domain, and together you can see that they look like a hand shape that has been described in many polymerase structures. And last but not least, it has this T-terminal domain, and here we have a zinc finger domain that is found here, and that is important for splitting the DNA during this process. So that's kind of an overview of the structure. There are a lot of things one can discuss. I'm going to highlight a few key points. So the first is how the enzyme remodeled the DNA during this process. So this is just a model of the DNA that we give it about 79 base pairs. And then here is what it looks like when it bound to this enzyme. I'm going to turn it around so you see the 2 arms of the DNA. So we take the protein away, and you see this is 1 arm, it's shown here, and then you tilt it around, you see the 2nd arm. So again, I'm going to put up the unbound DNA. So this part is the same, but this arm here needs to bend down more than 90 degrees in order to adopt this conformation during this process. So now let's zoom into the active site to see what we have there. So here I'm going to take the protein away to show in the nucleic acids. Here the top strand of the DNA is in dark blue, the bottom strand in light blue, and the RNA is in red. And the DNA is quite flexible. The regions of the DNA we cannot assign the sequence specifically, but you can see in the active site you're very clearly poly A RNA that is being duplexed with the DNA. And here's our cryo-EM density. So just to kind of recapitulate this point, so we literally go from here, which is the DNA we give it, and the enzyme take it and bend it to something like this so that it can start the synthesis, cDNA synthesis. So again, when we did the variability analysis of this interaction, what we found is that the DNA kind of thread through the enzyme back and forth like this. So one arm gets longer, the back arm gets shorter, so that it goes back and forth like this. It's very dynamic. And to our surprise, most of the interactions were DNA backbone. So there's not much sequence specificity as we kind of expected. And then what we also see, we see very little no base-specific interaction with the poly-RNA. Again, this is quite surprising. We were expecting a bit more sequence specificity. And last but not least is that, however, we look at the maps, whatever states we look at, this top strand that is kind of, when we give it as a full strand, always appear to be broken. You can see here, for example, this gap between these DNA segments. So as drawn, really deliberately here, that it is given like this, but in the density it always look like this. So this promises to ask whether the enzyme in this process also has cleaved the top strand. So how do we test this biochemically? To do this, we probe the reaction, the time course of reaction, and probe the top strand and the bottom strand. So we have our substrate, two labels now. So the bottom label is a Cy5 and the top label is a FAM. So what we do is we monitoring that synthesis of the bottom strand, as you've seen, and this is the time course. And then if we then monitor the same reaction using the different color on the top strand, as you can see, during the course of the reaction, we start to see smaller bands appearing on the top strand, suggests that this top strand in the process also get cut by the enzyme. Well, not yet, but the next question we ask whether the enzyme did it itself. And indeed, to do this, to confirm this is ORF2, that this is the reaction will make a mutant that cannot cleave this DNA. And indeed, we're monitoring the top bottom strand synthesis. You can see the reaction products here, but if we're now monitoring the cutting of the top strand, we see no products. So this suggests that during this process, it also cut the top strand, but cutting of the top strand is not a prerequisite for this reaction, a TBRT reaction to happen. This still can happen. So this kind of observation has important consequence for the sequence event that happened during retrotransposition. So in the introduction, I told you that the top strand is cut, the bottom strand is cut, first strand synthesis, and second strand, the top strand is cut. So this, based on our work, it suggests that the enzyme first cut the bottom strand and then cut the top strand, and then do the first strand synthesis instead of other way around. And if this first strand synthesis is then complete, then the second strand synthesis happen, you get a full length insertion. But if the enzyme then start to synthesize the second strand before this first strand is complete, what you have is truncation, actually 5' truncation. And indeed, most of the LINE-1 copies in our genome are 5' truncated, at least 90-something%. So this explains that the enzyme cut the top strand before it do the first strand synthesis. Therefore, it sometimes jumps to the second strand and starts synthesizing and explaining a lot of this outcome of these truncated LINE-1 insertions. So then, having confirmed that the enzyme cut the top strand, we asked where are the cut sites. So in the patient that we took the sequence from, the cut site is right here. So again, as you can see from the gel, there are 3 major bands. So we asked whether these coincide with this patient, whether at least one of them coincide with this cut on the patient DNA. So we turn into an old school technique. We are here. This Sanger sequencing was pioneered here by Sanger. So what we just run Sanger sequencing and identify specifically these 3 cut sites. So the first cut site we identify is right here. second cut site is right here, and third cut site is right here. So none of that match what was seen in the patient. And this is actually not quite surprising, because what we have the in vitro system, but in cells what is known as these elements really co-opt a lot of cellular factors to its own advantage. So probably we are missing the cellular context of this reaction here. So around the time with this in mind, AlphaFold 3 just literally came out. And so we thought, okay, that we know that probably many cellular factors involved, but whether we can use this tool to see whether we can find, based on our structure work as well as previous functional work, any cellular factors. So we did an AlphaFold screening of cellular factors and found 2 high confidence factors that form high confidence interaction with LINE-1. One is PCNA, which is normally involved in DNA replication. It's a processivity factor for DNA polymerases doing replication. And this really nicely places the PCNA in the line of the DNA that is in our structure. So this go through the central channel PCNA that normally accommodate DNA. So the second factor we also found from the screen is a poly-A binding protein cytoplasmic 1. And again, here we found 2 RMs of this PAPC1 protein, and this places the RNA in the right direction compared to what our structure has in the active site. And I'll tell you a little bit more about how we validate these interactions. So just to kind of, again, to see how PCNA is predicted to interact with ORF2, so that's the interaction sites. And what we found is, compared to previous structures of PCNA interacting partners, which I'm showing here, so these are the peptides of previous proteins that have been shown to interact with PCNA. You can see that they are kind of binding a very similar way, except for this red thing that is actually the prediction we have of this ORF2. So it binds in a very different way from normal PCNA interacting factors. And George is American, and he likes going to the pub since he come here, so he thought of naming something to remember his time in the UK, so he named this PC unusual binding motif, or the pub motif. So it's to his credit for this naming. So then with this, we then set out to look into more closely how this interact. So it uses the PUB helix, which we call the PUB, but it's actually a helix for multiple interaction with PCNA. And interestingly, previous work that has performed alanine scanning mutations on ORF2 and look at how these mutations affect its retrotransposition activity, we found that the residues on this PUB helix is critical for its activity, because if you mutate that, you really abolished LINE-1 activity when assays in cells. And furthermore, this PUB helix is very highly conserved, especially among mammalian LINE-1. And so with this in mind, we then mutate 2 of these residues in this motif, a tryptophan that is here, and also an isoleucine that is in the back in this schematic here. So we did a pull down with PCNA with ORF2. So we have ORF2 tagged and immobilized, and then we boost PCNA, and you can see the wild type pull out PCNA robustly, but the mutant does not. So this validates this interaction is probably happening in the test tube as well. So next, we then turn into our other interactor, which is a PAPC1 protein. And again, as I mentioned, it really plays the polarity of the RNA in the right direction compared to our structure. Again, we found that PCNA is PAPC1 is predicted to interact with ORF2 using a helix. And George, again, English-loving American, named this the PAPC1 interacting essential element, or the pi element. So again, this is also, these elements also very important for LINE-1 retrotransposition activity, as you can see here in this diagram where we map how these alanine scanning mutations based on the previous work. Again, this is only very conserved in the line, as you can see from the sequence alignment. So we tried to do this pull down, as I showed in the previous slide, but this protein is actually PAPC1 is very sticky. The results are not always conclusive, the bit of background binding. So we then turn into a different technique, which is immunofluorescence. And thanks to the help with the people in the live microscopy facility here, where we then express ORF2 in human cells. And normally when you do that, you have this puncta of ORF2 in the cytoplasm. And then we also stain for PAPC1 in the cytoplasm. You can see here in the wild type ORF2, you can see this co-localization of PAPC1 and ORF2 in the cytoplasm. But if we make the mutations at this pi helix, you can see you lose this co-localization. And this is then quantified by Jerome from our LINE microscopy facility. So where we think this interaction might be important, as I introduced earlier, ORF1 and ORF2 like to bind its own mRNA. And what you can imagine is this own mRNA can be bound by PAPC1 in the cytoplasm, and as ORF2 being translated, it can bind this own mRNA co-translationally via this interaction with this PAPC1. This is, in the field, this is a property called cis preference. And this is quite famous. I could explain potentially how this enzyme like to bind its own mRNA. So with that, I will summarize. So here, what I presented to you is we capture this LINE-1 ORF2 with a pre-nick RNA target on DNA, and that allow us to see how the DNA is remodeled during this reaction that you can see in this cartoon here. We also discovered that during this process, prior to cDNA synthesis, it also cut the other strand, and this had important implications in the order of sequence events that happen during retrotransposition. We also, through AlphaFold modeling and also biochemical validation, we found potential interaction between ORF2 and PCNA, as well ORF2 and PAPC1 protein, and these proteins might be important in the life cycle of the LINE-1. So with that, I will stop and highlight and thank the people in the lab as well, highlighting George. George started his lab at Princeton and are looking for postdocs to join his labs. If anyone here likes his work, do reach out to him. And Hongma also helped George on the way, and our colleagues in structure studies and all the facilities here for all their support. And thank you for your attention.
Brilliant. Do we have any questions in the room?
So when you did the dynamic analysis of the DNA, you suggested that it was threading through, but couldn't it just be that some parts are, in some particles it's more mobile and some particles not, in different classes rather than, say...
Yeah. The movie is more between the states, so I guess it's yeah, but I think there's a bit of going back and forth too, even in the sample, because one, it's open and it cuts, the enzyme can go, because we also have 3 cut sites. So yeah, it's we don't have if we have a single molecule to capture this, it was cool, but we don't. So we only see it by morphing between the states.
Any ideas about the loop that it creates that's absent, but that will be largely single-stranded disorder?
Yeah. So it's I guess a limitation of this is, you know, what is this disorder you can't see, and that's where I think being here and maybe talking more of you know, the single molecule crown where this can be captured dynamically would be cool. So in EM, you kind of, with structure, what you see is snapshots and you kind of morph between it, but you lose the time context. Yeah.
Very great talk. So with your new factors identified based on the models, can you now sort of explain the cut site that's observed in cells?
Yeah, it's a great question. So yeah, we were very surprised to see very little specificity. So there's a couple of things. So basically, LINE-1 is quite opportunistic, right? So it's normally acts during replication. So PCNA could be helped to bring it to wherever the site is more, you know, kind of exposed or something like that. But currently it's not clear. It's still not clear. But I think it's more complex than just PCNA. I think it's probably hijacked replication, and PCNA happened to be there. And it's also, yeah, it uses PCNA to probably hijack replication, but the site specificity, I don't know whether it's defined by PCNA or another factors in cell, but it's still, I guess, to be elucidated, yeah. But we were surprised that it's really in vitro, there is not much specificity yet.
Maybe building a little bit on that. So I was quite curious about the AlphaFold piece. You found these predictions that were high confidence with 2 different proteins. How many candidates did you have overall? How clear was that?
Yeah. So we actually we didn't do extensively because you remember when it came out, you can only run 10 jobs a day. So we have multiple people making accounts and logging in. So there was a limitation in that.
Yeah.
So what we did is we look at the literature and look at mass spec of factors that's being pulled down with the LINE-1 from cells, and then we narrow our hits down that first, because then I think the increase is to 20, but it was still not enough to do like a proteomic wide. But potentially now I think the code is released, so one can imagine doing a proteome wide screen. Yeah.
There's a question online.
Question online from Maddie Pronker says, fantastic talk, Kelly. Since ORF2P couldn't cleave the DNA bottom strand by itself in your reconstituted system, how do you think this happens in people?
Great question. So we did come back to that. So basically what we found is that so we just being naive and doing plan end, but if we have a single-stranded end on the 5' end of the DNA, then actually it cleaves fine. So there's a bit of a substrate requirement that plan end is not the best substrate in hindsight, but we only found out in the revision. So of course, yeah. But now knowing that we need to provide, and that's also again going down to replication, where it's acting. It's really depend if it's probably not acting when it's all chromatinized and all closed. It's probably acting when the chromatin are open, and there's probably this single-stranded DNA that might be created during replication, for example. So that's something we found out during the revision. Yeah.
Thank you very much, Kelly.